Okay, welcome to Brother Bailey's very entertaining video series. Um, Today we are going to talk about taste, mostly because stuff tastes good. Think about a chocolate bar, all the delicious tastes that come with that. Chocolate in and of itself, of course, is bitter. You've got salt, you've got sweet. All of these things blend together to give you just that perfect chocolate taste. And it's our job today to understand how to get from stimulant to action potential. And this is something that I mentioned previously. The brain only speaks action potentials. And as we understand special senses, our job is to understand how to convert something in the environment to an action potential. All right. So with that said, let's see if we can draw a tongue and understand some of the surface anatomy before we get into the signaling. Okay, there's a couple of things that we can see as we look at how the tongue is divided up. First of all, and we saw this when we looked at cranial nerves, the tongue is actually innervated. When it comes to taste buds, the tongue is innervated by two different cranial nerves. And this division can actually be seen here with this structure right here. This is called the foramen cecum. And is in most of us just scar tissue at this point. This is the attachment point for the thyroglossal duct, and this is formed during the embryonic descent of the thyroid. So as the tongue, I've got two different parts of it. As it comes together, the thyroid actually drops right down between the two of them, and then the tongue fuses, and you have this little scar tissue. Typically closed at birth. There are some people around there who actually have this remain patent or hollow, and then they have a hole going through their tongue, which is kind of cool to look at. What that gives us, though, is a border where we can see the posterior 1/3 and the anterior 2/3 of the tongue. The anterior portion of the tongue, this is innervated by cranial nerve 7, the facial nerve. And the posterior, I said third and row half. The posterior third is innervated by cranial nerve 9, glossopharyngeal. Okay, as we start thinking about taste buds, it's important to recognize that taste buds exist on what we call papillae. And there are 4 types of papillae or papillae, depending on where you're from. That we can find on our tongue. The first one, I've actually drawn these in blue right here. These are called circumvallate. Which essentially means to surround with a rampart or a defensive wall. And if we were to draw this, it might look like this, where this actually isn't sticking out, but there's a big, huge groove or remote that's surrounding it. And so here's our circumvallate papilla, and we need to recognize that the taste buds in here would be here, right? Where food is going to get kind of trapped down in that moat, and the taste buds will have the opportunity to survey it and assess it while it remains down in that groove. The second type of taste bud that we would like to talk about is a taste bud found on the sides of the tongue. Excuse me. A papilla found on the sides of the tongue containing taste buds, and this is called a foliate papillae. Foliate just means leaf-like, right? So these are thin sheets that kind of look like leaves. And these are ventral folds on sides of the tongue, 4 to 5 folds per side, and they are covered with lots of different with lots of different taste buds. The other one we can mention is fungiform. Fungiform is kind of a mushroom shape. You can throw in here. These are fungiform. These actually would extend above the surface, kind of like a big mushroom. And this is where you'd find your taste buds. Um, Oftentimes, in fact, you can do this if you have a little circle of paper. You can take it with a specific diameter, stick it on your tongue, and then dye with a blue dye everything that's in there. And that blue dye causes these taste buds to pop out, and you can count the density of taste buds. Everybody has different densities of taste buds. There are super tasters out there who actually insure their tongues because it's their livelihood. These are going to be your professional wine tasters who can taste something and immediately distinguish all these different sorts of tastes. It just has to do with the density of taste buds on your tongue. I'm more kind of the guy that just says, give it to me as long as it tastes mostly good. I'm going to eat it and not worry too much about it. There is one more type of papilla, but this type of papilla doesn't have any taste buds on it. And this is the Filiform Papilla. We can draw those here. Filiform, no taste buds. So what is it used for? This is to increase friction to allow you to roll bolus of food around your mouth. So this is simply all about friction. It is also important to notice, I think, that when we think of the whole idea of taste, this refers to a rather complicated process. Not only do we have activation of taste buds, and we'll talk about that in just a minute, but we also have to think about smell. We have to think about texture. Can't tell you how many times kids say, oh, I don't want to eat that because it just feels funny, or it makes my teeth squeak, right? All of these things go into how we interpret this idea, this rather abstract idea of taste. So activation of taste buds, smell, texture, pain is another one that is super important. And this actually works independent of taste buds, but if we're thinking about hot peppers, ghost peppers, or something like that, right? We can actually register these on a pain scale where capsaicin is something we like in spicy foods. Some people like spicy foods, some people don't. It just, again, lends to the idea that there's more than just the activation of taste buds that contributes to taste. And this is going to be evident in a minute because especially in stark contrast to smell, where I've got over 900 different receptors for 900 different odorants, and then the combination of all of those things could give me the ability to distinguish between well over 1 trillion different smells. With taste, I'm kind of limited, right? And I think you've all probably played those games where if you plug your nose and blindfold, you can't hardly tell the difference between an onion and an apple. So there's a lot that goes into understanding how taste works. But for us, we're just going to explore now taste buds and their various activation. Okay, let's zoom out. Let's look at this. Let's bring up another screen. When we think of taste buds, I want to recognize that per taste bud, there are between 50 and 150 different receptor cells. And I know probably in elementary school they talked about, oh, you taste sour on this part of your tongue and you taste sweet on that part of your tongue. You might have biases for one or the other, but for every taste bud, you've got representatives of all 5 different receptor cells, and there are a lot of them. So when we think about these receptor cells, what do we have? We have sweet. Well, let's do this. We have salt, and we have sour, and then I'm going to change color. We have sweet and bitter and something called umami or savory. And it'll make sense in a minute why we color coded them that way. But I want to go back to this idea of a taste bud, right? And we zoomed into one of those taste buds on one of the papillae. So if we look at our papillae, right, we've got this idea where if we go to Circumvallate papillae, these taste buds, for example, are hanging out right up here. And if we zoom into that, we get a structure maybe that looks like this. This is not by any means anatomically correct, but within it, the point is this. We have these receptor cells, maybe a couple of blue ones, and there are 5 different colors. Maybe a green one. Let's add maybe a couple of yellow ones. Orange one. And a red one. So within this taste bud, I've got 50 to 150 different receptor cells. And I just want to take a minute to appreciate from a big picture point of view what needs to happen. Each of these, based on color, I guess, so the blue, these will tie into each other. Of course, green will be a completely different neuron. Yellow is going to be a completely different neuron. These are my first order neurons. And this is going to be the cranial nerve. So we already talked about cranial nerve 7. The facial nerve, we talked about cranial nerve 9. Glossopharyngeal nerve. In infants who also have taste buds associated with their epiglottis, this would include the vagus nerve, cranial nerve 10. But as we get older, those taste buds disappear. if we follow all of these back, where are they headed, right? These cranial nerves. We know that these nerves, in particular, facial and glossopharyngeal, they're going to plug into the nucleus tractus solitarius. So they're going to synapse on what we would call a secondary order neuron, and this is going to be in the medulla oblongata. And we are looking for, well, I don't know if you need to know this or not, but it's the nucleus tractus solitarius. I write that just to highlight the idea of what a nucleus is, right? We talked about a ganglia being a collection of cell bodies outside of the CNS. A nucleus is a collection of cell bodies inside the CNS. So it's fun to see that. Now, where do you think we're headed next? Remember, except for smell, all sensory is going to be shunted into and processed by the thalamus. And so that's where we are here in our tertiary order neurons. This is in the thalamus. We call that the sensory sorter. And where do you think it's going to be sorted to? It's going to go to lots of different places, but one of the primary targets is going to be the gustatory cortex. Think Chef Gusteau on Ratatouille, right? Gustatory has everything to do with taste. And this is going to be found primarily in the insular lobe of the cortex. So that's the signal transduction pathways that are going to communicate in the form and language of action potentials to the parts of the brain that are going to register this. What we're really interested in is what's going on right here. Because here we have something a little bit unique. We have a receptor cell communicating with a neuron. Why is that so different? Because the receptor cell is an epithelial cell. And that's what makes it different. So epithelial cells, these are not designed to communicate via action potentials. And yet we know why it's so important, because think about the last time you ate a hot bowl of soup or drank a super hot cup of hot chocolate. And you scalded your tongue. You've essentially destroyed your taste buds. It might be 2 or 3 days before those taste buds come back. But that's the point. They come back. We can't do that with other sensory, even in the best with the neurons that we talked about in the olfactory sense of the smell. These neurons turn over, it takes weeks to months. And going without taste for months simply because you ate something spicy or hot is really not acceptable evolutionarily speaking. So we need to have a cell type that has the capability of turning over very quickly and replacing itself. And epithelial cells do this. We see it in the skin, right? The skin's constantly turning over. We see it in the gut. The gut's constantly turning over. So epithelial cells have this innate ability to divide quickly and rapidly, and that's what allows us to keep fresh taste buds, these receptor cells, on the surface of our tongue. But it does pose a problem. How do we get epithelial cells to activate neurons to initiate an action potential? All right, to look at that, to explore that, let's come back up here and look at the two different classes essentially of receptor cells. We have salt and sour that are grouped in the same because of what these things actually bind to, or what it is that triggers them. For salt, the thing that triggers these taste buds is sodium. And for sour, the thing that triggers these taste buds, think Sour Patch Kids, it's actually acid. It's hydrogen ion, H+. Now, this is really convenient because both of these just happen to be cations. So this allows us the simple ability, if we look at one of these receptor cells, all we have to do is have a specific cation channel on the surface of the cell. Let's say it's for sodium, right? And then we're tasting salt. Well, in comes the sodium through a leak channel, and when sodium comes in, what happens? All of these positives, these positive ions build up inside the cell, and that leads to cell depolarization. So very similar to a neuron, we actually have embedded in the side of this cell a voltage-gated calcium channel. And as soon as the cell depolarizes with the introduction of sodium, calcium comes into the cell. And based on everything we know, remember, calcium, this causes exocytosis. Now, this is a little bit different because when we think about the vesicles being exocytosed, remember, these are not neurons, so they are not making common neurotransmitters. In fact, what's actually released is ATP, and ATP is being used as a neurotransmitter. So on my primary order neuron, if we were going to draw that, so here's my neuron, and it has what's called a P2X ligand gated cation channel. And the ligand for these P2X channels happens to be ATP. So that when ATP binds, this channel opens up. That's going to lead to sodium, and it's going to lead to calcium. These are going to enter the cell. And now, because we're talking about neurons, that's going to lead to your standard depolarization, threshold action potential. We're off and running. We're communicating that action potential to the brain through the pathway that we discussed just a few minutes ago. Okay, what about these other three? These, if we're looking at sweet and bitter and umami, sweet, this is glucose, right? Dextrose. If we're talking about bitter, we're talking about quinone. We're talking about umami. We're talking about amino acids, specifically glutamate, that will bind to these. Of course, these are not ions, and that prevents a little bit of a problem because it was easy to allow cations into the cell and cause depolarization. But that doesn't work here. So for these three, we have to use G-protein coupled receptor signaling in order to get it done. So let's come over here and see what this would look like. Okay, just as a quick review, we've seen this in the video, you've seen it before in other G protein coupled receptors. We start with a 7-pass transmembrane receptor. It is associated with alpha subunit and beta gamma, and the job of beta gamma is to tether alpha if alpha is bound to GDP. But along comes a ligand, maybe it's glucose, right? Glucose comes and binds to this G protein coupled receptor. And the job of the receptor, once it's activated, pull out its scissors, cut off the GDP, and replace it with a brand new shiny GTP. That liberates alpha subunit because once alpha subunit is bound to GTP, beta gamma can no longer tether it. So now we've got alpha subunit bound to GTP, free from beta gamma to go activate something. And in this scenario, the protein that's activated is a protein called back up on that, phospholipase C. And the job of phospholipase C is to cut a phospholipid on the internal leaflet of the membrane called PIP2. If you want more details on that, you can go watch the video. But we'll draw this. There's my 2 fatty acid chains, glycerol backbone, and then my phosphate group and my polar head group, and it's got some phosphates on it as well. This guy is named PIP2, phosphoinositol bisphosphate. The job of phospholipase C, once it's activated, is simply to cut. So he pulls out his scissors and cuts off this structure right here called IP3. And once IP3 is liberated, if you remember from that video, IP3 is going to come down and it's going to bind to this ligand-gated channel on the surface of the endoplasmic reticulum. And we know what's going on in here. The job of the smooth ER, one of the things that it does is to store calcium. And IP3 binds to a ligand-gated calcium channel on the ER, and this allows calcium outside of the cell. Or excuse me, not outside of the cell, but into the cytoplasm outside of the smooth ER. This calcium is going to do a couple of different things. I'm not sure it's important to understand. It's going to open up TRPM5, which is a calcium channel that's going to let in extracellular calcium. But regardless of all of that, this is going to lead to the release of ATP. In this case, it's a little bit different because we're actually not exocytosing it. This is the non-vesicular ATP release through a protein called calcium homeostasis modulator 1. It's a voltage-gated ion channel tied to ATP release. I don't know that it's super important to recognize any of that, but the story is let's let calcium out, and this time we're dealing with primarily intracellular calcium stored in the ER, and that calcium is going to cause the release of ATP. ATP is my neurotransmitter. That will activate the cranial nerves, the neurons associated with either the facial nerve or the glossopharyngeal nerve. And now I've got action potentials going to the brain, and I'm registering that as
taste.